Okay, so this week we're going to do a couple different things. We're going to start out, we're going to do a biofilm assay, and then we're also going to identify bacteria that you've already isolated from various soil samples using polymerase chain reaction of the 16S ribosomal RNA subunit. We're going to send that out for sequencing, and then we're going to get the sequence back, and we're going to do a phylogenetic analysis on it to see which organisms you have. All right, so we'll start out with biofilms. So what is a biofilm? So biofilms are simply communities of microorganisms that grow on a surface. Now, you can often have more than one microorganism in a biofilm. What we're going to work with today, we're going to have a single organism in each biofilm that we form. But they grow, you can see here, in aqueous environments. So we can see the sort of organisms just growing on these rocks, and there's microorganisms there also. If you don't clean your shower that often, you get something that looks like this on a shower curtain. You see some mold growing on there. So that's a biofilm. It's growing on a surface. It's a microorganism. This is a knee implant structure, and you can see this biofilm that is grown on that. And then one that people don't think about a lot, but affects us every day, is dental plaque, biofilms growing on your teeth. Okay, so how do biofilms form? Now, what we normally think of bacterial cells growing is we think of this. Individual cells, like shown here, swimming around in some sort of liquid. Now, when we grow them in the laboratory, this is how they usually exist, because we put them into flasks in liquid, and then we shake those flasks or the tubes, whatever it may be, and there's this vigorous shaking which prevents cells from attaching to any sort of surface. Now, out in the real world, there may be no such agitation, and cells can, when they come in contact with a surface, can attach to it. They will adhere to a surface. This initial contact nucleates more cells attaching, and as they attach, they secrete substances, polysaccharides. Sometimes there'll be proteins in there, and some cells die, and the DNA gets all sort of mixed up in this sort of meshwork, this kind of mucousy layer that sort of holds them together. As the time progresses, these biofilms will grow, and you'll see three-dimensional structures that come out of them. You can see in there water channels, and it's sort of shown here, where you can see all the cells sort of stuck together with the sort of this polysaccharide material holding them together, and then you'll see these channels in between. This is a neat way of looking at bacteria. These are cells where the DNA has been stained with DAPI, and it will fluoresce blue. And then eventually, the biofilm will grow and be very dense, and some cells are going to be released, and this can be triggered by an environmental factor based on what nutrients are available, and these cells will be released, and they can. So if we look at this as sort of, with a sort of microscopically, so this is a confocal scanning laser microscopy, so we can start seeing sort of this sort of nucleation event where cells start to adhere to the surface. And eventually they will cover that surface. Then you start to see these sort of bumps start growing here, and they will continue to grow, and until you get these really sort of large mushroom-shaped colonies that form. So you can actually observe this with the right kind of microscope. So biofilms are of huge concern. Now, you don't need to know this list here, but this is to give you an idea that there are lots of human infections that involve biofilms. And I mentioned dental caries, periodontitis that happen in our mouths, but there are lots of different kinds of infections. These are also, biofilms are also important with other kinds of surfaces. They cause biofouling of equipment, and they can damage equipment if you get biofilms forming on them. The other thing is that biofilms are highly resistant to antibiotics. So when we're looking at infections, this is really, really concerning. So they have a slower growth rate. Antibiotics cannot penetrate into them because they have now this three-dimensional structure. It's tough to get through this extracellular matrix that they make. And they also express genes that increase their tolerance to the various stresses, including antibiotics. There are other features about biofilms that make them more resistant to antibiotics, make them more sort of more hardy. And so I've shown a very similar slide to this previously, but what we're going to focus on is what's actually going on in this extracellular matrix. So cells, as they start to accumulate in the biofilm, are not all identical. Now, even in a biofilm that's formed with the same bacterium, in sort of lighter gray here, if you can see it, are things that are called persister cells. Now, these are dormant cells, and we're going to talk about them and how they help a community of microorganisms resist stresses. The other thing to note about the biofilm is that there's an oxygen gradient. So oxygen has a difficult time penetrating deep into this biofilm because of all this extracellular matrix and all the cells sort of crammed together. And antibiotics have the same issue. So this sort of structure of the biofilm just really makes it resistant to lots of stress. I find persister cells to be just fascinating. Right? So persister cells survive killing by antibiotics. So if you look at a population of cells, right, we have these sort of normal cells in blue, and the persister cells are shown here in red. Now, you treat with an antibiotic, the persister cells survive. These blue ones, they die off. If you now grow in the absence of an antibiotic, the population will regrow, but you will now get a combination of persister cells and sort of normal cells. Now, why is that? That's because the persister cells, this is not a genetic change in the persister cell. They are still the same bacteria as these blue ones. It's just that they have, for some reason, become metabolically inactive. And so when the population regrows, the same sort of fraction of them that were persister cells remains. This is different from resistant cells. So in resistance, in a cell population where resistance appears, here we have these sort of normal blue cells, and then there's one purple cell that's resistant. This resistance is now due to a genetic change. This cell is genetically different. It has some sort of mechanism to resist antibiotics. So when you treat this population with antibiotics, these blue normal ones die off. The resistant one remains. When this regrows, either in the presence or absence of antibiotic, it's going to have a genetic change in it that gives the bacterium resistance to antibiotics. So this population that comes from this cell, they should all be resistant because they're going to have the genes just to resist the antibiotic. That's different from the persister cells, in that only a few cells will be resistant to the antibiotic. Okay, so we are going to set up our biofilm assay using a 96 well plate format. So we're going to grow cultures in these 96 well plates without shaking, because remember, we do not want to shake them because we want to give them an opportunity to adhere to the walls. Then we're going to wash out the cells that have not adhered. So after a day, we'll just put them in water and wash them out. Now the cells that remain bound to these wells, so these little circles here, we're going to stain them with crystal violet, the same stain that you use in gram staining. We're going to wash out the crystal violet after a certain amount of time, and then we're going to treat these wells with acetic acid. Now the acetic acid is going to extract the crystal violet from the cells on the wall. The cells remain bound to the wall. Just the crystal violet is going to come out, and so you'll see a darkening of the liquid in the well. And so in wells like sort of shown here, like these four here, that have may have had a lot of cells bind to the walls and form a really nice biofilm, well, they're going to get stained more with more crystal violet. And so when we extract the stain with the acetic acid, we will get a darker purple. Now, in the cells adjacent here, some cells have adhered to the wall, but apparently not as many as have adhered in these wells. And so these will get stained to a lesser extent because there's fewer cells there. When we extract the crystal violet, it'll be a lighter shade of purple. And so we can quantify this whole plate by putting in a spectrophotometer, which you have used, except now we're going to use a particular spectrophotometer called a plate reader, and it can read all these wells at once, and it will give us basically numbers of how much purple, so we can quantify this. It's really nice. So the more purple, the more cells that adhered. Okay, so we are going to set up our plate in a very systematic way. We're going to use 4 media, LB with glucose or with sucrose or with both. We've used these before for our growth experiment. I like these sugars. And we're going to do it in such a way that when we look at our 96 well plate, we have our 8 well side and our 12 well side. For each of the lettered ones, we're going to put a different media. So we're going to put in the top one, we just put LB across all of these wells, these 12 wells. And we're also going to do that, count down 4, in row E. And so we're going to have the same media in total of 24 wells sort of separated into these different zones. Now we're going to add bacteria. So we're going to use 7 different bacteria and a no bacterial control. And so they're going to be arranged in these sections of 12. So these sort of blocks here. And so these will get all the same bacteria in this light blue area. So now we'll have bacteria 1 that's being grown in LB, LB plus glucose, LB plus sucrose, and LB plus glucose and sucrose. All right. And we're going to do that for each of the different bacteria. So we set up our plate by putting the media in first in all the appropriate wells. We'll go row by row, and then we'll add our bacteria. And when we've done this, each organism will have been done in triplicate, including our control where we don't add any bacteria. So this will be our negative control. Now, if you look at sort of these blocks, so we'll look at just this sort of row H here. These are all going to have the same media and the same bacteria. So these are going to be replicates of each other. But these are going to be technical replicates. So what does that mean? Technical replicates are repeated measurements of the same sample that represent independent measures of the random noise associated with the protocols of the experiment. So what does all that mean? It's basically how well have you pipetted everything? Is everything working well? So it's sort of checking on you and the equipment that we're using. Now this is different from biological replicates. So if we did this same organism 5 here, but we repeated this block more times with just organism 5, each time the starting material for the organism comes from a separate colony. Well, now those are going to be biological replicates. They are parallel measurements of biologically distinct samples that capture random biological variation, which may itself be a subject of study or a source of noise. So we could have set this up where we take bacteria 1 and just do that 4 times, and bacteria 5 here and do it 4 times, but we're not. Okay. So another thing that we're going to do in this during this week's lab is we're going to determine what one of your organisms is that you isolated from soil sample or from somewhere else, where you can use this pretty much in any sort of way that you like. And we're going to amplify the 16S ribosomal RNA gene using PCR. We're going to determine its sequence, and then we're going to use phylogenetic analysis to figure out what it is. So I'm going to start talking about the phylogenetic analysis part. So what is phylogenetic analysis and why do we perform it? So there are two major components. One is phylogeny inference or tree building. This just gives a branching order and evolutionary relationships between taxa. So taxa is a word. These are just can be genes, they can be populations, they can be species. And this is what we're going to use it for. You can also do character and rate analysis. So you can use the phylogenies that you make to get an understanding of how certain traits evolve. Okay, so some common terms that we use for phylogenetic tree analysis. So here's a typical phylogenetic tree, and there's some interesting features that you should know. So we have our taxa. Again, these can represent pretty much anything for us. They will represent the 16S ribosomal RNA gene. And the singular of this is taxon, so we'll have A through E. Now, these are called terminal nodes. These are the ones at the end of the tree. These are the ones that exist now. They are the ones that are currently alive. Internal nodes, or these divergence points, represent hypothetical ancestors of the taxa. These are no longer alive. We can't see these, but we can infer what the sequence are. If we're looking at different genes, we can infer what the sequence is at each of these points based on what they are in the taxa that still exist. And so this point down here is the ancestral node or the root of this tree. And these are called branches, all these lines here, how the tree branches. Now, we can divide these branches up into clades. So here's one clade of A, B, and C. This is also a clade of B and C, which is internal to this clade of 3. D and E also form a clade. And if you want to look at it, all of these can form a clade, depending on how you look at this. Okay, so this is just another way of looking at phylogenetic trees. What do these sort of distances all mean here? Like how the length of these and how far these are apart going vertically. Well, first of all, vertically here has absolutely no meaning. Right? The spacing is just whatever makes it easy for us to see what's going on. Right? The order in which they appear is just randomly given. It's how they're all connected that matters. So this sort of blue dimension that I've written here is important. Now, in cladograms, it's not important. There's no scale to this. They're just showing how the organisms branch. In phylograms, the length of these bars is proportional to genetic difference. So these are more closely related, D and E are more closely related to each other than B and C are because they have a shorter distance here. And in ultrametric trees, it's proportional to the time. These are true evolutionary trees. This could be something in millions of years ago. So another way of looking at this is a tree is a set of nested parentheses. Right? B and C are more closely related to each other, but yet these two are more closely related to A. So we have this sort of configuration here. D and E are more closely related to other, but they are still related to the other 3. And so we just write these out as in sort of parenthesis form. And this sort of explains that, what I just said. So the data that we're going to use for building our trees is a character-based method. Now you could base this on the length of a femur, or how many bones are in a hand. But we're going to look at DNA sequence. You can also do this with protein sequence, and we're going to infer this tree sort of branching order from these sequences. So here we have our species A through E. And here's a small region that has been sequenced, and it's shown. And you can see at some positions there are differences. Right? So there's a couple of A's in A and B, and then there's T's in the rest. And you can see like here's G and then C. And so along this, you'll find that there are differences. So how do we use these differences? So we can do, for distance-based methods, we can do pairwise comparisons. And we can transform this sequence into numbers, so into distances of dissimilarity. So we have 20 nucleotides here. There are 4 differences out of 20, so that's going to be 20%, or 0.2. Right? So we do this for all of them, and we get this matrix. Right? So this is comparing A to A, there should be no differences, obviously. And we just spoke about for A to B, right? This 0.2 is coming from here, and we get all these different distances, and we can use this to then. How do we obtain these sequences? So the sequences are in the bacteria, and we need to get large quantities of DNA from those bacteria to sequence. So we could grow up lots and lots of bacteria and isolate their genomic DNA, or we can take a colony of bacteria and subject it to polymerase chain reaction. And so we're going to use PCR to amplify the 16S ribosomal RNA gene. Now these PCR products are going to be sent out for DNA sequencing, so we're not going to do it ourselves. We're going to send it out, and we'll get the data back in a couple of days, and then we'll take that data and we'll use a BLAST database for ribosomal RNAs. Now, we're using the 16S ribosomal RNA gene. Why are we using that? So this RNA is part of ribosomes, right? It's in the small subunit of the ribosome, right? And this is the part that helps to decode the mRNA, helps to set the reading frame. It forms base pairs with the Schindel-Garneau or ribosome binding site sequence in the messenger RNA for bacteria to tell the ribosome where to start translating. Now, the large subunit, which is not going to be involved in any of our analysis today, but what that does is that catalyzes peptide bond formation. And so the larger ribosomal RNA actually has the catalytic function for forming peptide bonds. So the ribosome is a ribosome, very similar to self-splicing introns or hammerhead ribozymes, which you may have heard of. So the function of RNA in the ribosome is critically important. So here's a structural diagram of the small subunit of the ribosomal RNA. In blue, you can see proteins, and orange is ribosomal RNA. And you can see, if you look at this, that most of this is actually made up of RNA. Now, that RNA can be written out in a two-dimensional structure showing regions where we get intramolecular base pairs, so base pairs within the same RNA molecule. So it shows these sort of like stem structures here. And then regions that don't base pair, which you have these terminal loops, and then you have these bulges. And this is when you take the sequence of ribosomal RNA from multiple organisms, you can sort of fold them up based on base pairing into these kind of structures, or into this structure. So why is the small subunit ribosomal RNA gene so useful, this 16S ribosomal RNA? And it's because it has both conservation and variation. So what do we mean by that? So this line here just represents the 16S ribosomal RNA gene from the 5' end to the 3' end. And you can see in blue are regions that are conserved. That means that if you were to get ribosomal RNA gene from different organisms, it's very likely that the sequence will be very similar, if not identical, in these portions from almost any bacteria that you isolate. So this is really good. So now we can design a primer for PCR, and we'll talk about primers in a moment. And when we talk about PCR, that will be able to bind to this sequence from pretty much any organism. So that's what these green arrows are the primers for PCR that go on one strand that recognize the top strand, and the reverse primers are showing an origin going in the opposite direction. Okay, so that's why conservation is good. Now, why is variation good? Well, if this ribosomal RNA gene was absolutely identical in all organisms, then you would amplify it, you would sequence it, and you get the exact same sequence from every single organism. You can't tell them apart. So you need some sort of variation. It allows us to tell the bacteria from each other. And so having this, so many variable regions, the more variable regions you have, the greater resolution you have. But usually we can identify to the genus level. Species is kind of difficult. Now, for our experiment, we're actually going to use this 27F and 1492R primers. So that's going to give us pretty much the whole ribosomal RNA gene, which is over 1,500 nucleotides. Now, there's also a large database of sequences from ribosomal RNA genes, so it's really easy to identify organisms using this, and this has become sort of the. So, polymerase chain reaction was this was invented by Kerry Mullis, who passed away in 2019. But he made a statement that I just sort of resonated with me. I'll just read it. It says, most people in molecular biology today are not old enough to remember pre-PCR, but try to do your job without it, and you will see what a difference that simple little technique has made. And this is true. So when I started, PCR had really just started coming into being used ubiquitously. And I think about the things that I've had to do. If I couldn't use PCR to do them, it would be near impossible. It's not impossible because people did them, but it would be very, very annoying. So this revolutionized recombinant DNA technology. So what is PCR? And so in a very, very sort of broad sense, it's basically making copies of a specific piece of DNA in a tube, so in vitro. And doing this very, very quickly. Now, this piece of DNA that you're interested in may be represented only once in a chromosome, and so you have it's in small quantities. You may not have much DNA material to begin with, and this allows us to get lots and lots of a piece of DNA. And so here we have just a picture just showing you a chromosome. So the chromosome, imagine, is a very, very large chromosome, and we just want this orange region. So we can design primers that sit just adjacent to this orange region and amplify this region and get more copies of it. But we go through multiple cycles and we keep doing this, and we get so much of this DNA that we. Okay, so I mentioned that PCR is DNA replication. It's in a tube. And so what do we need to do this? So we need a template DNA. So for us, it is going to be a colony of bacterial cells. Each bacterial cell has DNA inside it. And through the PCR, the cells will break open and they'll be able to access that DNA. But it can be genomic DNA that's been isolated. It can be soil samples that we extract the bacteria and then extract the DNA from them. There can be lots and lots of different places. We're going to need primers. These are also called oligonucleotides. These are going to be complementary to sequences at the end of the fragment that we want to amplify. So that orange region that I showed in the previous slide. And they're usually pretty small, between 15 and 20 nucleotides. We're going to need deoxynucleotide triphosphates. So if you remember from DNA replication, these are the components that get incorporated into a growing DNA strand. So we're going to provide those. And we need a DNA polymerase. And we're going to use a thermostable DNA polymerase called Taq DNA polymerase. It was isolated from the bacterium Thermus aquaticus, and its name comes from the Taq, the T is from thermus, and the Ac is from aquaticus. And it will become very apparent in the next couple slides why we need it to be thermostable. Basically, we're going to heat up the reactions multiple times. And so when setting up our PCR in the lab, these components are going to be contained in a premix that's they're already mixed together, and we're going to add to that our template DNA and our oligonucleotide primers. So what goes on in PCR? So each PCR cycle has 3 steps. It has a denaturation step, and that's shown here. So we start out with our template DNA as a double-stranded DNA. There's all these hydrogen bonds holding it together. We heat it up, breaking those hydrogen bonds, and the strands come apart. So this is denaturation. So we go to a very high temperature. We're going to then lower the temperature so that the primers can anneal. And so that's shown here. These little green arrows are the primers. And then we're going to raise the temperature, and we're going to extend that. So this is the temperature where the polymerase works best at. And that's these sort of pink regions here. So after we've gone through one cycle, we start with 1 copy, and then we duplicate that. We end up getting 2 copies. And this is what the cycle looks like as a temperature profile. So this is how long roughly each part of the cycle takes. On the X, on the Y, are the temperatures, and you can see we have very high temperature to denature. We lower that down to allow the primers to anneal, raise it to temperature that's optimal for the DNA polymerase, so it can synthesize DNA, and then we're going to repeat this. So this is 1 cycle, and then we repeat this. So if we look at the repeat of this, now in the next cycle, this double-stranded portion that has just been synthesized will get denatured and can act. We now have both these strands can act as templates for DNA synthesis, and we will get 2 more copies here. However, we also have this other copy that was synthesized. That will get denatured, the primers can anneal, and we can get DNA synthesis of those new pieces. So now we have 4 copies. If we go through 3 cycles, we will have 8 copies. And so this will, theoretically, the amount of PCR product will double every cycle. So after 30 cycles, you have 2 times 10 to the 30, or 10 to the 9th copies of your DNA. Okay, so back to just mentioning the primers again that we're going to use. So remember, this is our ribosomal RNA gene, and we are going to use this 27F primer, which will be on 1 strand, and this 1492 primer, which is on the reverse strand. So you need to have 2 primers. They need to both have their 3' ends facing towards each other, so that we can have DNA replication occur on 1 strand this way, and then 1 strand this way. So after we amplify these, we'll run them on an agarose gel, take a look at them, and then if they're good, we're going to send them out for DNA sequencing. So how does DNA sequencing work? So DNA sequencing is another version of DNA replication in a tube. So the method that we're going to use is the Sanger, or what's known as the dideoxy sequencing method. And we're not going to perform this ourselves. We'll have a company do this for us. But this is basically taking advantage of DNA replication. So we'll provide a template DNA strand. Now this is in the form of our PCR product that we generate. We'll provide a primer for sequencing. Now this will be the same primer that we use in our PCR, right? So this will be one of the same primers. And what we want to do is we want to determine this sequence that's here in that's shown here. This will be the sequence of the ribosomal RNA gene. So the primer is 5' on the left and 3' on the right, and so this can be extended by DNA polymerase. Now the DNA polymerase also needs the deoxynucleotide triphosphates, so we're going to give those. Right? We're going to provide those, but we're going to provide something additionally that will allow us to determine the sequence of this strand. And what those are is nucleotides, but now they are called dideoxynucleotide triphosphates. And we'll have 1 for each of the nucleotides that we're used to. And then each one of those is going to have a fluorophore attached to it that's a different color. So when the chain gets terminated, we'll be able to detect which nucleotide was the last one incorporated. Okay, so why are these important? So you may remember from structure of DNA. It uses a sugar called deoxyribose, and it has a hydroxyl group on this position. This is known as the 3' carbon. This is normally where the incoming nucleotide will get attached. So the phosphate that's on the 5' carbon of the incoming nucleotide will get covalently linked to this position, forming a phosphodiester bond. Now, by using dideoxynucleotide triphosphates, they don't have this OH, this hydroxyl group. It's just a hydrogen. Any incoming nucleotide cannot be attached to this. So once this dideoxynucleotide triphosphate gets incorporated, polymerization stops, and we'll proceed no further. Okay, so what happens? So we mix all these guys together. So if the first nucleotide that gets incorporated after this primer across from this G happens to be a C that is a dideoxy, you'll get incorporated, and that will stop the extension of that of the newly synthesized DNA strand. So it's going to be the size of the primer plus 1 nucleotide. Now, the ratio of these dideoxys to the deoxynucleotide triphosphates is important. We have to have a lot of these and very few of these, so that most of the time the normal nucleotide will get incorporated. So here we'll have a C incorporated. So that's going to extend it by 1 nucleotide. But then if, again, by chance we get the dideoxy version of this T, which will be opposite of this A in this position, it's incorporated, we stop synthesis here. This molecule is 1 nucleotide longer than this one. And so this proceeds for each position. So we'll get normal synthesis, and then a dideoxy gets put in. Normal synthesis, and then 1 base later, we'll get a dideoxy put in. So what you can see is that we have now a bunch of different molecules that each differ by 1 nucleotide. Now, at the 3' end of those, the nucleotide, the dideoxynucleotide that was incorporated, has a fluorophore. And so they have different colored fluorophores for each nucleotide. So these products get separated by electrophoresis, and then as they run through the gel, they have specialized equipment. It's a capillary electrophoresis. As the fluorophore passes by a certain point and has a laser, this will get excited. It will emit its appropriately colored light. That light will can get detected, and so we can determine the actual sequence of these of nucleotides. And in this case, it would be it's given here. Right, so you will get to see something like this when we get the sequencing back. This is the fluorescence that has been measured, and it's this chromatogram. And so you can see if we pick this first position, we can see that as the nucleotide approached the detector, we could see the fluorescence start to increase until it gets to its peak when it passes right in front of the detector, and then it starts to move away. It goes back down. So what you see is a succession of peaks that are shown here. Some are so large that they go sort of off the scale here. They sort of get cut off at this point. But you can see each of these peaks, and you can just simply read the sequence. Here it would be A, G, and there are 2 C's and A. So you just read the sequence like this. And this will be the 5' end shown on the left and the 3' end on the right. What you also see on here is if you look down at the bottom, you see all this sort of other stuff. Now this is noise. The DNA molecules may not be perfectly made. The primers may be missing a nucleotide. So you'll get a lot of noise. But that noise should be far lower than the signal that you get from most of the sequencing reaction. Now the height of these is not critical. They just need to be above this sort of background level, and they have to be distinct enough that they can be determined. Okay, so that's sort of winds up what we're doing for this experiment. We're going to take DNA sequence that we're going to generate by PCR and DNA sequencing. Get that DNA sequence back. We're going to take it and plug it into BLAST, and we're going to get phylogenetic data, and we're going to see what our organism is that we've isolated, and how it matches up with other organisms in the database.